Hi everyone, this is going to be a little bonus video lecture that will just be for your reference after class today and not a homework thing. So if you're watching this, hopefully it means you want to be watching it and it's not because I forced you to watch it. So in this video, we're going to go through the different different types of systems that cells use to keep their proteins well folded and not aggregated. And we'll also be looking at a couple selections from different papers that have to do with our different sorts of cellular protein quality control mechanisms. So the one that we're going to start with is this chaperone and chaperonin types of molecules. So these are basically assistant helper type molecules that can make it or increase the likelihood that a protein is going to fold correctly. And there are different types shown here. So the difference between a chaperone and a chaperonin. So chaperones are these smaller types of molecules that will help newly forming or nascent polypeptides fold correctly by binding to regions that may be at risk for misfolding. So you can imagine that as a protein is coming off of the ribosome, if, for example, there's a region of hydrophobic amino acids that don't like being around water right here, right now, there's no other hydrophobic regions for these amino acids to bind to. So they're at risk because if there's a protein that they're not supposed to attach to, or if there's other hydrophobic stuff, they're so desperate to get away from the water that they might attach incorrectly. So the chaperone is going to come in and just sort of like hold that region until its binding partners or other hydrophobic regions can come in, and then that would be the appropriate thing for them to interact with. So chaperones are just these smaller molecules that attach onto newly formed polypeptides and sort of just hold them until everything's made. And that way the protein is able to fold correctly once the whole thing is created by the ribosome. And then chaperonins are these barrel-like structures that open up on the top, and polypeptides are able to go inside of them to get a sort of safe environment where they're protected against any sort of negative influences that might be hanging out in the cytoplasm. So it gives this really secure and safe environment for the polypeptides to fold up. And then once the polypeptide's folded, the chaperonin will open up and it's able to exit, and then a new polypeptide can come in to be folded. So it's sort of just a safe space for polypeptides to go into in order to be folded. Okay. One example that we're going to be looking at in the next slide of a chaperonin is called a heat shock protein, or HSP. And heat shock proteins got their name because the cells make more of them if a stress like heat is applied. So stressful conditions can increase the amount of chaperones and chaperonins, but they also have to do with neurodegenerative associated proteins in this next figure. Oh, nope. Sorry, I was... Okay, so here we are going to be investigating the role of a chaperonin, HSP70. So HSP70 is just one of those chaperonin barrels in a fly model of Parkinson's. So in this paper, what they did was they were looking at dopaminergic neurons, which are neurons that express dopamine, and visualizing them using this brown staining. So what we can see is that in this control picture, we've got 3 dopaminergic neurons on the left and about 4 or 5 over here on the right. And what we're looking at is how the number of dopaminergic neurons changes over time. And the setup looked like this. So they are using flies, and so these neurons are in flies. And in these flies, they expressed either one of 2 things. Some of the flies, they made them express alpha-synuclein, which is a protein that aggregates and is associated with Parkinson's disease. And in other flies, they expressed alpha-synuclein, but they also expressed HSP70, this heat shock protein, or chaperonin. And so in these pictures, you can pretty clearly see that the flies that just were made to express alpha-synuclein, this dangerous or Parkinson's-associated protein, have fewer neurons. So their neurons are dying. So there's only 2 here. But if they had the flies express that alpha-synuclein, but also express the chaperonin, HSP70, then you can rescue against that cell death. So these flies still have a lot of neurons left, even though they're still expressing alpha-synuclein. And this is quantified over here in this graph. So on the y-axis, we're looking at percentage of dopaminergic neurons that survive. And so right at the beginning, it doesn't really matter if they're just expressing alpha-synuclein or if they're expressing alpha-synuclein and the chaperonin, HSP70. But over time, so you can see after 20 days, the neurons that only have alpha-synuclein in this light purple color have died by almost 50%. But the flies that got alpha-synuclein and the chaperone and HSP70 still have close to 100% survival. So this shows that HSP70 is able to protect against neuronal loss in this fly model of Parkinson's. Okay, so our next system is the ubiquitin proteasome system. And in this system, we are dealing with, again, a way to sort of handle misfolded or damaged or messed up proteins. And the way that we handle them is by attaching a tag onto the molecules. And this tag is going to send the molecule or the protein to the proteasome. And the proteasome is just a big garbage disposal for proteins. So the protein is gonna go in and they're gonna get spit out as just like little pieces, maybe single amino acids even. And so not just any protein can go into the proteasome, you have to be sent to the proteasome. So you have to be targeted to the proteasome. And the way that you get targeted to the proteasome is with a molecule called ubiquitin. So ubiquitin is just this little tiny molecule, and it can be attached onto a protein, and usually it's a chain of them. So you get a chain of ubiquitin molecules onto a protein, and that signals to the cell that this protein needs to be destroyed. So if the proteasome encounters a molecule that has this chain of ubiquitin, it will degrade that protein. And so that's the ubiquitin proteasome system. Ubiquitin is the tag, the proteasome is the garbage disposal. So in this paper that we're going to look at in the next, or in the figures that we're going to look at in the next slide, I want to make one point. So within a cell, ubiquitin is attached by different enzymes, and the ubiquitin is made within the cell itself. But scientists are also able to make a sort of man-made ubiquitin tag and attach it onto any protein that they want to. So if we make like a synthetic ubiquitin chain and put it onto a protein, it'll still work to send that protein to the proteasome. And we call that synthetic ubiquitin chain a degron. And the name even sort of makes sense because it's a degron tag, and the purpose of it is to signal to the cell this protein needs to be degraded by the proteasome. So a degron tag is a synthetic ubiquitin chain that should send that protein to the proteasome. So what we're looking at here is a cell line that has a couple things going on. So in these cells, they've expressed a mutant cystic fibrosis protein. So all of these cells have mutant cystic fibrosis proteins in them, and these proteins are prone to aggregation and misfolding. And they've used this tag of a flag, which it's not really important what that tag is, but essentially it just allows us to visualize where this mutant cystic fibrosis protein is. So in this panel, we can sort of see, but there are a bunch of cells here, and there's one particular cell that has an aggregate. So you can see this bright spot, and that's all of the mutant cystic fibrosis protein aggregating in this one area. And they've pointed to that cell with this white arrow. In this exact same group of cells, there's another molecule being expressed. And so in this exact same group of cells, they've also expressed GFPU. And what this is GFP, which is green fluorescent protein, and it's just in black and white here. It's green fluorescent protein that's been tagged with a degron. So that's the exact same molecule that I was just talking about in the last slide, this fake ubiquitin chain that should still send whatever it's attached to the proteasome. So under normal circumstances, GFP with this degron should be going to the proteasome. It should be getting sent to the proteasome to be chewed up. And if the GFP is chewed up or sent through the garbage disposal, it can't glow anymore. GFP's ability to fluoresce has a lot to do with its structure. So if it gets chewed up, it's not going to glow anymore. So we should have GFP being made, and as soon as it's made, it's getting tagged with this degron, this synthetic ubiquitin chain, and then it's just going to get sent to the proteasome and be degraded. So what you're seeing in this panel is this really faint amount of fluorescence in, for example, all of these cells. And the fluorescence is really faint because the GFP is getting destroyed as soon as it's getting made, because it has that degron tag. But you'll probably notice that there's one notable exception to the faint glowing, and that's happening in this cell. And it happens to be in the exact same cell where we have an aggregate of the misfolded cystic fibrosis protein. So in the same cell where the mutated aggregated cystic fibrosis protein is clumping up and aggregating, we're seeing that the proteasome isn't able to degrade the GFP. So this suggests that when there's aggregation in a cell of proteins, protein aggregation in a cell, it inhibits the ubiquitin proteasome system from working properly. So this cell where there's an aggregated cystic fibrosis protein isn't able to degrade the GFP with the degron. And that is presumably because the proteasome isn't working properly. So the take-home message from this paper is that aggregates can inhibit the ubiquitin proteasome system from functioning, and we can measure the health of the proteasome by using this degron fused to GFP. And these are just quantifications of what we showed in the picture. So cells with a lot of misfolded cystic fibrosis protein have higher amounts of fluorescence, which means that the GFP is not being degraded or the proteasome system is not working. And then the same thing over here, cells without aggregates compared to cells with aggregates. And we're comparing them on the x-axis, we have log fluorescence. So again, you can see higher fluorescence in the cells with aggregates. And this higher fluorescence means that the cell is not degrading GFP, even though it should, because the GFP has that degron attached. Okay, take a breath. That one was kind of a lot. We're going to move on to autophagy. And remember that autophagy is this system where we're actually able to sort of bulk degrade whatever we want to degrade. So autophagy works for organelles, it works for proteins, it works for aggregates even. And so the way that it functions is that we're going to enclose whatever we want to degrade inside of a membrane, inside of a vesicle. And so we form this membrane that encloses everything we want to get rid of. And then we attach to that or fuse with that vesicle a lysosome. And you maybe recall from the cell parts exploration that we did, the cell structure election, that the lysosome is responsible for degrading a lot of the cell's material. So inside of a lysosome, there are a bunch of enzymes that will just chew up whatever it encounters, these little Pac-Man enzymes, because they're going to eat up whatever's inside of, they'll eat up anything. So when we fuse the lysosome with this vesicle, the lysosome enzymes are released, and then these enzymes are just going to chew up whatever's inside of it and will be released with just the products. So proteins will be broken down to amino acids. This mitochondria will be broken down to its component parts. So we can use autophagy to break down stuff as well. And that might include misfolded or partially misfolded or aggregated proteins. So in this figure, this paper, we're looking at the relationship between the proteasome system and autophagy. So in this first panel, we're looking at, in green, a marker for autophagy. So this is just baseline control conditions. And you can see that there's a little bit of autophagy happening, like a baseline amount. But in this left-hand or right-hand panel, they've applied a drug to the cells, and this drug inhibits the proteasome from functioning. So remember, the proteasome is the garbage disposal with ubiquitin, and they've made it so that that system as a whole can't function. And as a result, or like when we do that, we see that there's an increase in the amount of autophagy that's happening in these cells. There's a lot more green, so there's a lot more autophagy. So if you inhibit the proteasome, you get more autophagy. And so it seems like autophagy and the UPS, ubiquitin proteasome system, are able to compensate for one another. So the cell's like, oh, I can't use the proteasome system. What else do I have at my disposal? Oh, I can use autophagy. So autophagy is upregulated or increased in response. And we've got graphs that just sort of quantify this over here. So we're looking at induction of autophagy, and this is baseline, or like what we're comparing everything to. This is where we're inhibiting the proteasome. So you see an increase in autophagy when you do that. And this is just a positive control that we know. It's a drug that we know induces a cell to undergo autophagy. So we should see an increase here. And then finally, here we're kind of going in the opposite direction. So if you inhibit the proteasome, you see an increase in autophagy. But here we're looking at what happens to proteasome activity if you inhibit autophagy. So this is a drug that prevents autophagy from working. And if you do that, you can see an increase in the amount of proteasome activity. So what we can see from these figures is that autophagy and the proteasome system are compensating for one another. If you inhibit one, the other one is going to go up. And if you inhibit the other, then the other one is going to go up. So if you inhibit autophagy, the proteasome system will increase. If you inhibit the proteasome, autophagy will increase. Okay, last system slash context here. So this last one has to do with oligomers and aggregates. So remember that when we have a protein, there's a native fold, which means the proper 3D shape, and then we also have misfolded shapes. And if we make sort of like small little clumps of these misfolded proteins, we form what are called oligomers. And these oligomers are still soluble. They're still able to travel around the cell. They're just small clumps of misfolded proteins. But if we keep putting these clumps together into bigger and bigger clumps, we can form different kinds of aggregates. So we can form disordered aggregates, we can form amyloid beta aggregates, we can form different kinds of aggregates, which are insoluble and a lot more immobile, but a lot bigger inside of a cell. So there's some controversy about, depending on the disease, depending on the protein, depending on the context, is it the misfolded proteins themselves, or maybe the misfolded oligomers that are still soluble and able to travel around the cell? Is it these things that are toxic, or is it the aggregates themselves that are the problem? So this last paper was aiming to look at this. And so we're going to look at two figures from this one. So in this paper, we are looking at cells that are all expressing the Huntington's disease protein. So it's HTT, and it's fused to GFP, just so we can keep track of how much of it is there and sort of where it is. And one quick note, in this paper, they use the phrasing inclusion body, but that's essentially, for our purposes, the same thing as an aggregate. So inclusion body equals protein aggregate. And so in this first figure, they're making a correlation between the amount of Huntington's protein that a cell has and how long it takes for that cell to form an aggregate. And what you can see is that if there's a lot of Huntington's disease protein present, aggregates will form more quickly. So after only like 40 to 50 hours. But if there's less Huntington's protein present, so if we're down here on the x-axis, that's associated with a longer time to form an aggregate. So something more like more than 75 hours. So the more Huntington's protein there is, the faster an aggregate is going to form. And there are two ways to interpret this finding. one way is like, well, of course the aggregates are going to form more quickly if you have a lot of Huntington's protein. Like there's a lot of bad protein and it's going to form aggregates because that's what happens. So it's unavoidable, but it's just a consequence of all of this messed up protein, and it's bad for the cell. An alternative way to look at this same data is to say that, that the formation of aggregates is actually protective, so that the cell sort of sees, oh, I've got all this Huntington's disease protein. How can I protect myself? I'm going to form an aggregate early, and that's going to protect me from all of this messed up protein. So the idea is that maybe stuffing all of this bad protein into one space, sort of sequestering it away, could be helpful for the cell rather than harmful. So a proactive, beneficial response. And in this, we're looking at neurons that either do or don't form inclusion bodies and tracking how likely they are to die over time. So we're looking at neurons. They all have this Huntington's protein inside of them. And at the beginning, you can see that everyone is the same. But then right before 75 hours, some of the cells formed a protein aggregate and some of the cells didn't. And so they separated those two out, the cells that did and the cells that didn't, and tracked them over time and looked at how likely they were to die. And you can see the cells that didn't form an aggregate in black and the cells that did form an aggregate in white. And what you can see is that over time, the cells that didn't form an aggregate are more likely to die. They have a higher risk of death. And the cells that did have an aggregate had a lower risk of death. So they did a lot more experimentation in this paper to sort of do different controls and make sure that the amount of protein was the same in these different cells. But the takeaway point here was that at least in this context, forming an aggregate, forming an inclusion body seems to be a protective response rather than something bad, rather than something pathological. So there's still controversy over the role of aggregates, and they play in the role of aggregates and what they're doing in all of these different diseases. Okay, so that's this video. I hope it helped solidify your understanding of these different protein quality control systems and the papers that we discussed in class. And I'll see you guys soon.